Okay, we are in our last phase of photosynthesis, and that is dealing with the Calvin cycle. We are going to take those products that we made in the light reactions of photosynthesis, the ATP and the NADPH, and we are going to use them to take some building blocks, some carbon dioxide molecules, and we're going to use those energy products to build those carbon dioxide molecules into a big complicated glucose molecule. Okay, so let's get started. It is the Calvin cycle, but as you can see, it is not this Calvin. It's called Start the Slideshow, Coleman. Alright. But indeed it is another guy, that Calvin, who came up with this Calvin cycle, who figured out that it existed and how it worked. Please remember your basic photosynthetic equation, which we keep going back to over and over again, but it's really important. In the light reactions, we figured out why we need water to make photosynthesis work. We need to strip electrons out of that to replace electrons that are lost from the chlorophyll A molecule. We know why we need light energy to excite those chlorophyll A molecule electrons in the first place, and we know where the oxygen is coming from. It is coming from the breakdown of this water molecule. The oxygen is released as oxygen gas. What we have not yet used or figured out is where the carbon dioxide comes in and where the glucose comes in. Okay, so we're going to look at that now. Okay, our light reactions basically are giving us ATP and NADPH. Both of these are forms of energy. This is a form of energy in that ATP. Remember the P's are 3 phosphate groups, and the last one in the chain has a whole lot of energy. If you break that bond, energy is released. The NADPH is carrying any energy-rich electrons, and we can use those. So what are we going to do with them? How are we going to use them? We are going to use this energy to build stuff, just like you would use the energy of your body to build a Lego tower. Okay? Photosynthesis, using light to make things, in this case to make glucose. Okay, so getting from this small, simple, low energy building block, CO2, to a glucose molecule. CO2 has very little chemical energy stored in it. Okay, remember, chemical energy that is stored in the form mostly of carbon-hydrogen bonds. You break these bonds, you release energy. Carbon-oxygen bonds have next to no energy because the oxygen is such a greedy hog of electrons. Once they have laid claim to some electrons in a covalent bond, you can forget it. You're not going to get them away from oxygen. So this is not itself got very much potential energy, but we are going to convert it to a molecule that does. Okay, CO2 is fully oxidized. The glucose molecule here is fully reduced, meaning it is holding a whole lot of energy-rich electrons. So we're going to go from this thing that has been stripped of all its usable electrons to this that has got lots of usable electrons. Okay. Now, in order to do that, it's an endergonic process. We're going to have to put some energy into taking our small carbon dioxide building blocks and making them into this big complex molecule. Okay, the action of getting from here to there takes many little steps. It is a biochemical pathway, and each step is catalyzed by an enzyme, and each step uses some of this energy that we made back in the light reactions. Okay, so the Calvin cycle is occurring in the stroma. That's this liquid space in the thylakoid, that surrounds the thylakoids. This whole thing is a chloroplast. This is a granum, which is a stack of thylakoids, and the Calvin cycle is occurring in this liquid space around the thylakoids. Okay, And we already said we need the ATP and the NADPH to make it go. Okay, the main point of the Calvin cycle then is to take carbon in the atmosphere, which is in the form of CO2, and use it to build a molecule of sugar. Our requirements then are CO2 from the atmosphere, that's our raw materials, and the energy that we get from the light reactions, okay, ATP and NADPH, okay, which we got from the light reactions. Alright, there are 3 phases to this Calvin cycle. The carbon fixation phase, the reducing phase, and then a regeneration phase. And we'll talk about those and you will understand each of them by the time we are done. The carbon fixation phase. I want you to think of carbon dioxide in the atmosphere as a beautiful butterfly that is fluttering merrily in the meadow. What our job is, if we want to fix carbon so that we can use it to build glucose, our job is to grab that butterfly, slam it down, and nail it to a board so it cannot fly away ever again. We want to fix it. We want it to be stuck to something that's going to not enable it to leave. Okay? This is the carbon fixation phase of the Calvin cycle. Essentially, there are certain chemicals that are able to grab carbon dioxide that enters the leaf through the stomata and attach them to larger carbon compounds so that it will not leave. It will be used to actually make sugar. Okay? So, the molecule that is going to act as the hammer and nail to attach it to a larger molecule is called rubisco. It is in fact an enzyme, even though we don't have the ACE. This is an abbreviation of a larger name that ultimately does have ACE in the ending. I'll look it up sometime. I've looked it up before. I can't remember what it's called. But rubisco is the hammer and nail. It is the thing that is going to grab the CO2 out of the atmosphere and nail it down to a bigger carbon compound so it can't fly away. Alright? The board, the big carbon compound that rubisco nails the CO2 to, is called ribulose bisphosphate. Ribulose bisphosphate, or we're going to call it RUBIP for short. Okay, RUBP, ribulose bisphosphate. So this is the board. Okay, rubisco is the nail, and we're going to nail the poor little butterfly to the board, and it won't go anywhere after that. Okay, RUBIP is a pretty big sugar molecule. It's a 5-carbon sugar molecule, so like a pentose sugar molecule. So it's pretty big. And what happens actually when we add CO2 to it, it actually would turn that into a 6-carbon molecule. If we have a 5-carbon, a 5-carbon molecule, and we add the 6th one that comes from the CO2 to it, what happens at this point is it's going to split into 2 3-carbon molecules. We really don't care about the name of it. I'm just telling you that that's what happens because it'll make sense when you see the diagram in just a second. So what we really care about is now CO2 is in a form that the Calvin cycle can use to make sugar. Okay? It is now in a usable form. So here we go. In order to make this work, please understand these numbers here are telling you how many CO2s, how many RUBIPs have to be involved. So 3 CO2 molecules have to come in. This is the atmosphere out here. So this is bringing CO2 into the leaf from the atmosphere. Rubisco nails CO2 molecules to 1 each to 3 different RUBP molecules. Okay, so we're gonna, this is a 1, 2, 3, 4, 5 carbon molecule. We're gonna add this to it, make it a 6-carbon, and then that's gonna split it into, if we've got 3 6-carbon molecules, ultimately we're gonna cut them in half, so we'll have 6 3-carbon molecules. I do not care that you know all that. What I care about is that you know that CO2 is coming in from the atmosphere. Rubisco is the enzyme that's going to attach it to this foundation molecule, RUBP, and then we're going to get something that the cell can work with to make sugar out of. Okay? Now, that is carbon fixation. We've caught the carbon. Okay? And we've nailed it down. The next phase is to stuff some energy in that molecule so that we're building a sugar molecule with all of its stored energy inside. Okay? So, all of that energy that we got in the light reactions, okay, we're now going to finally use it. Okay? Phosphate groups get transferred from ATP. So we're going to take ATP molecules and convert them to ADP and inorganic phosphate. And we are going to use it. We're going to stick these phosphate groups on to those molecules we just made when we fixed the carbon. Okay? That stores energy in those molecules. We are also going to use the NADPHs. We're going to take their electrons. So NADPH is going to become NADP plus again, because we're going to take its electrons and we're going to stick them onto that molecule that we made when we fixed carbon as well. And the product is going to be a sugar called G3P. Note that G3P is not glucose. G3P is not glucose, but it is used to make glucose. It is the precursor to glucose. Okay, so here is that in picture form. Alright, so here is what we made when we fixed carbon. Now we are going to add some phosphate groups to it, and here you can see we're adding phosphate groups to it right here, and we are going to give it energy because we're adding these energy-rich phosphate groups. Additionally, we're going to take some electrons. Remember, electrons always travel with hydrogen ions as well. So we're going to take these electrons from the NADPH, and we're also going to stick them into this compound. And what we're going to end up with is 6 molecules of G3P. This is a sugar. It is not glucose, but it is important because it can be used to make glucose. Okay? Now, please note, G3P by and large is going to get just recycled back into the Calvin cycle. Only 1 of 6 that we make here is actually going to be sent out to make glucose. Alright, what happens to the rest of it? Well, it gets recycled. This is the regeneration of the carbon dioxide acceptor that we know as RUBIP. We've got to recycle RUBIP so we can grab more carbon and keep doing this process over and over again. Okay? So, as we already said, we've just made 6 molecules of G3P. 1 of them is going to be sent out to make the glucose. The other 5 are going to go back and recycle the RUBP molecule. Okay? We need a little bit more ATP to do this, so again, it's an energy demanding process. We need a little more ATP. So if you look, here's our 1 G3P that came out and is going to go make glucose. The other 5 are going to go up. We're going to add some ATP, 3 of them, and we're going to make 3 molecules of RUBIP, which are going to go and accept 3 molecules of CO2 from the atmosphere. So we complete the cycle and we're ready to go again. So if you'll note, that's 6 ATPs, 6 NADPHs, 3 ATPs. It's a ton to make 1 G3P, and if I'm not mistaken, I believe it takes like a couple of these to make a glucose. So bottom line is this is very energy expensive. It is very energy expensive. Lots of ATP, lots of NADPH, but we have a virtually unlimited source of energy. And what is that source of energy? It is light, and so that's why this works. Okay. All right. So, okay. 3 turns of the Calvin cycle are going to give us 1 G3P. What that means is it takes 3 CO2s from the atmosphere to make 1 G3P. but it takes 6 turns of the Calvin cycle, here you go, here's the math of it, to make 1 glucose molecule. Okay, so we've got to do a doubling of all the numbers we saw back in that diagram to make 1 glucose molecule. Okay, so I don't think anybody's going to ask you about this. The bottom line is it takes a whole lot of ATP and a whole lot of NADPH to make 1 glucose molecule. Okay, so I think this is just kind of saying all we've talked about already. Yeah, and this doesn't do it any better, I don't think, than what we just talked about. We don't need any more than what we've already had. I do want you to know that G3P, we've seen it before. Do you remember in glycolysis where we sent glucose into glycolysis and we do some stuff to it and we end up splitting glucose in half? Well, the half that goes into the rest of glycolysis is G3P. Okay, so it's not hard to imagine G3P kind of going through a reaction to become glucose, because when we break down glucose we get G3P. That's just a little sideline. All right. No, we don't need that. We don't need that. I think we are done. This is all summary. I think we can summarize in class pretty well. Yeah. All right, I think that'll do it for us. Thank you for watching, and we'll catch you next time.